Hey, boys and girls. We are live. What's up?
Hello, hello. Yeah, sorry I was a little bit late joining. I was cleaning my room and wasn't paying attention in time.
Hey, Walker.
Congratulations on finishing the semester, Walker.
Yeah, it's kind of cool. It's nice to be done, and it's also terrifying that I'm now a junior.
So, check private chat.
Okay.
Good question for y'all.
Who's Professor Evo? Oh, is everyone changing their name to Professor except for the actual professor?
Yeah. Yeah, we're actual professors. Yep. You got the joke. Anyway, so we're here to talk about genetic entropy, and one particular conversation I think that you had with Godzilla Freak and SFT the other night, I think.
Yep.
And I listened to a bunch of it. So, and you had an idea about how you wanted to proceed, Andrew, or did you?
Yeah, I mean, my thoughts on it were just Dan can share basically a quick rundown of why GE is bogus, and then we can kind of get into the specific responses that SFT and Godzilla Freak gave to me the other time. So.
Yeah. So I sat through that whole conversation. So
Dear God.
You're welcome. It was painful. It was a litany of all the talking points with no logical connection between them. It was just like, so let me just do a very brief, like, what is GE and what are like the problems? And then we could talk about that conversation specifically if you all want. So let's start with genetic entropy, which is John Sanford's idea. This is Dr. John Sanford, plant geneticist, and he wrote a book called Genetic Entropy and the Mystery of the Genome. And it has since been through several editions. I think I have the second edition of the book. I don't know what they're up to, but there's at least a third edition after the one that I have that is slightly different in a couple of places. But basically the idea is, Sanford's claim is that within the genome, you have the inevitable accumulation of harmful mutations that have effects on fitness that are small enough that selection cannot remove them. So over generations, you have this constant accumulation of very weakly deleterious mutations, and that over time results in a population-wide decline in fitness, ultimately leading to extinction. That's the idea of genetic entropy. Now, if you read Sanford's book, which the funny thing is, a lot of people that like to talk about genetic entropy have not read Sanford's book, but I've read
You don't have
Sanford's
to call me
book.
out like that.
Well, no, I wasn't calling you out. I was calling creationists
It's first on my
out.
reading list. Don't worry.
I'm calling creationists out for being like, here's the genetic entropy argument. I'm like, okay, here's what he says on page 25. Can you like, and they're like, well, I don't have the book. Like, okay, you haven't read the book, but you're going to tell me I'm wrong about it. Great. Thank you. So this is the way that Sanford makes the argument. These are the requirements for Sanford to be correct, right? And if you're familiar with population genetics, you're going to see some problems here right away. So one is that you need to have, for every mutation, you have a fixed fitness effect. Every mutation is either good or bad. It either has a large effect or a small effect. And whatever that effect is, that's what it is. And that is going to be independent of context. right? It's just,
Okay.
it is what it is. And right away, we got a problem because mutations are context dependent, right? It depends on the environment. It depends on the selective pressures being imposed on the population. It depends on the population size in terms of whether or not that mutation is selectable or not, right? In terms of whether the effect are large enough. So right away, that's wrong. Then you have virtually no beneficial mutations. His distribution of fitness effects, or DFE, has basically an immeasurably small number of beneficial mutations. He says they are so small we can round down to zero, basically. And that's obviously wrong because we can document beneficial mutations. We can observe the fitness effect. Like, it's clearly not zero. It's clearly a countable, like it's a non-zero fraction are detectably beneficial in terms of their fitness effects. Then we need to have a constant accumulation of harmful effects of harmful mutations over generations. Now, the problem here is that this is in direct conflict with the assumption that they are fixed fitness effects. Because if you have, each mutation has a fixed fitness effect. Every time a bad mutation occurs, you're now changing that distribution. But Sanford describes an approximately constant accumulation over generations. And you can read his stuff and he'll talk about X number of mutations per generation. And whatever fraction of those mutations are going to be harmful each generation. Well, if we get 100 mutations every generation and 90 of them are going to be harmful, you're describing a linear accumulation of mutations that is mathematically incompatible with fixed fitness effects for each mutation. Because when a bad mutation occurs, you're removing that from the possible universe of future mutations, and you're replacing it with the reverse mutation, which necessarily must be beneficial if the original mutation is harmful. So, and we're not done. We're not done with problems yet, because he also ignores relative fitness within a population. For Sanford's argument to work, you need to have zero differences in relative fitness within a population. Zero selectable differences within a population. Everybody has to be getting worse in such a similar way that there can be no selection between the different genotypes that are present within a population. But we're not done yet. Because all of that stuff is premised on the wrong definition of fitness. Because Sanford is not talking about reproductive output, which is the correct definition of fitness. Sanford is talking about this in terms of, he defines it a couple different ways, but the information content in the genome or the functional information in a genome, there's a couple different phrases he uses. But he's talking about it he uses the wrong definition. He's redefining the term fitness to suit his arguments rather than using the correct definition, which is reproductive output.
I would say he doesn't define it. Because
fair enough.
If we're talking about population genetics, I need math. Please give me my math. And if you define it in terms of reproductive output, I know what the math is. I don't know what functional information means in terms of math, in terms of genetic entropy. So it's not even defined. It's just a word.
Yeah. So that's actually going to get into Andrew's conversation with SFT, because it starts with, here's a specific case, quantify this. And 5 minutes of hemming and hawing basically get you no answer.
Oh, you can't ask SFT for math. That's not fair.
Fine, but granted. So there's one more problem we need to mention here before we get into the specifics of this conversation. And the problem is that all of this assumes an ideal starting point, because the only way you can have fixed fitness effects for every mutation and virtually every mutation being harmful is if you start at an optimal point and then degrade from there. Now, the argument of genetic entropy, the whole point of it is to prove that is the case, right? That this degradation is happening. If you're starting from an assumption of an ideal genome state, and then over time, if that's your starting assumption, you're begging the question in the formal sense. You're starting with your conclusion and then using that assumption to draw the conclusion that you want, right? So that's the last big red flag problem.
I think we could also add the fact that even using the terms genetic entropy or even error catastrophe, like we were talking about before, like there's no observational evidence for it. So it's kind of, like you said, begging the question.
And I will say, if anybody wants to come at me with any one of the studies that people claim exhibits error catastrophe in viral populations or in other populations, bring it, because like that's literally the thing that I did in grad school, was use a mutagen to induce error catastrophe was the thing I was doing. So like I know all those studies chapter and verse, and I can tell you specifically why each one didn't demonstrate error catastrophe. And in many cases, the studies that creationists cite, the authors don't even say they demonstrate error catastrophe.
And thank you for mentioning that, because it reminds me, I can actually define genetic entropy better than Sanford does, because basically what he's saying is genetic entropy is the idea that error catastrophe or extinction vortices, or whatever you want to call it,
well,
apply
we'll get into that, because those are opposite. So we'll get into that, but those are opposite things.
But they apply at all possible population sizes.
Yes.
But just slower. That is what he's saying. He is saying that genetic entropy is the general case of these specific cases, and providing no evidence for that, obviously. But that's what he's saying. I don't know why he can't say it, but that is what he's trying to say, right? We already have this thing. We know if you have really small populations, the effectiveness of selection is reduced because the population size is very small, and you can accumulate a lot of harmful effects, and eventually that can be very bad. We know, we've seen it. We've seen it in a number of populations that get isolated, small populations. And yeah, sometimes they go extinct from that. It'll happen. You're talking about, though, 50 individuals, like an effective population size of 50.
Right.
Like, it's just, so the idea of it is just it's just ridiculous, because we know that this will not work on large populations. It just doesn't.
Right. We know like selection is important for even larger populations, for instance.
It's also worth noting that when you have an extinction vortex, the thing that's going on in the population genetics of that population is the opposite of what genetic entropy would claim, right? Because genetic entropy is extinction due to too many harmful mutations that break too many things. You're greatly increasing the allelic diversity of a population. You can't select out the broken alleles that are being generated via mutations. With an extinction vortex, you get genetic drift, you get inbreeding, and you get an increase in homozygosity. So you're decreasing the allelic diversity and fixing a bunch of recessive, harmful alleles. So you're losing diversity. It's, in terms of the actual like genetics of the population, it's the opposite thing. So you can't just be like genetic entropy, genetic entropy, genetic entropy, and then oops, extinction vortex. Like, wait a minute, those are the opposites in terms of like what's actually happening in the populations.
2 points. 2 points with that. One, if Sanford understood the science he was using, he wouldn't have come to the conclusion he has. And two, if we're going to stop at every point and say, well, here's why it's even more wrong, this is going to go until next week.
We're going to be here all night. Yeah, no, that's true.
Yeah.
It's worth noting that the thing that I just described, Sanford literally does that in the book in the span of 2 paragraphs. He goes from describing mutations accumulating, causing the population to lose fitness, and therefore the population shrinks. And then in the next paragraph, he goes, and then extinction vortex, lots of harmful traits that are recessive start being expressed. And it's like, dude, that's the opposite. And he does it like he just flips the switch within 2 paragraphs without seemingly noticing the contradiction.
So I guess at this point, should we get more into the nitty gritty of the conversation I had?
Let's do it. Do we want to pull it up, or do we want to just kind of go through? I mean, I took notes.
I made a list of responses, so maybe we could do that, because I don't have the patience to listen to the video over and over again.
Andrew, I do have a transcript. If there's something you want to search for, I can search the transcript. So
okay. All right, yeah, that might be helpful.
I pulled up most of the papers that SFT popped up, so I've got them up here, and I can display any one of them if you want. If you just give me the name of the or get some idea which paper. So
okay, all right. So I guess should I start reading from this list I have of all the things?
Yeah. So I have it starting with the bristles stuff. Is that where you're starting?
Yeah, that's most of what I got.
Let's do it.
Some of these are kind of similar to each other, so might be some overlap. But the first one from Godzilla Freak was, it's so I guess everyone here knows the example we're talking about. So do I need to go over it again, or should we?
The bristles thing?
Yeah.
Go over it for people in the audience who aren't familiar with it.
Okay. So based on this paper that was sent to me, basically there was this T to G substitution in this gene called SCOOT in a specific species of Drosophila, D. santamea, and comparing that gene sequence, or not the gene sequence, an enhancer of that gene, comparing those sequences to a related species, they found that this specific substitution probably caused a decrease in the number of bristles along the male genitalia, that specific region, and correspondingly, or I should say simultaneously, caused an increase in the number of bristles along the male forelegs, also called the sex combs, which I did look it up recently, and they do have an effect in related species on the male reproductive success, but I couldn't find if this specific example had any fitness effects, so that I should be honest about.
Mm-hmm.
So should I get into the claims now?
Yeah. So what did they? So you asked them basically. Here's the mutation.
Oh yeah.
Here's the situation. Is this an increase or a decrease in information? Right.
Right.
Was that the part?
Right. Yeah. Basically, is it a net loss or a net gain? And if it's a net loss, what specific part contributes to that loss? Because they have to say it's a loss, or at least until this discussion, I thought they did.
But they didn't.
Right.
Just kind of waffled on it.
Right, and that kind of gets into their first response from Godzilla Freak, which was basically comparing it to like hairs on humans, for instance, and variation in that, and saying it's a quantitative difference, or actually use the tallness in humans as an example. So it's a quantitative difference, but the information is the same, so nothing changed. That's the first response I recorded from that conversation.
Right. So like virtually all mutations are harmful according to this new standard that we've invented for what fitness means. Okay, here's an example. Does it apply? Not really, maybe, sorta. This one, it depends. Not so much, basically.
Right.
Like, okay. So there was several minutes of like going back and forth, and it was a lot of generalities and not giving a specific answer. And I think I see in there SFT got into the rust on a car analogy, and it's like, okay, great. Like that's not an answer to the question, right? It's not an answer to the question.
Well, like my thoughts on it.
General claim, here's a specific case. Do they match? And the answer was things degenerate in general. Okay, that's not an answer.
Right, like my thoughts along this were basically, okay, if the information content didn't change according to their words, basically, and this still had phenotypic effects, then at least hints at the possibility maybe information isn't something we really need for evolution to take place.
Maybe what matters are traits and their fitness effects, and not this amorphous idea of information in the genome. Yeah. That's what I take away from it. That's because, I mean, this is a good question you came up with, right? It's okay. You have this hypothesis. Let's test it against this specific case of a very well documented mutation. What's the answer? The answer is we don't know. The answer is the answer is they don't have the tool. Like creationists don't have the tools to be able to quantify this stuff, and that kind of then the whole idea kind of falls flat on its face if your whole hypothesis is based on this amorphous thing decreasing over time, but you can't quantify that thing anytime there's a specific example. Then what are we doing here? Right.
And this was kind of another response, which I think SFT said. So I'm going to say two of these actually because they're kind of related. The first one was there are too many low impact deleterious mutations. So this is just a localized, potentially non-deleterious change, but it doesn't fight the genetic degeneration. And the other thing would be all these simulations he talks about and how that's accounted for and whatnot.
So is he talking about Mendel's accountant?
Oh, he's talking about Mendel's accountant, and I'm so happy CRISPR is here because CRISPR was one of the participants in a peaceful science thread from December where people really dug into Mendel's accountant and found some really interesting things in the code.
It was so painful to read that code.
Okay, which version of the code did you read, Christopher? The Go version or the C version?
I think I was looking through both.
Okay, because I wasn't sure which version they're currently using. Like they did a build, and they have a build on their website that you can download, and I'm not sure which version it is. So.
I looked through the Go. I'm more familiar with how C works, so I think I spend most of my time with that. Okay. But it's just awful. I don't know where they learned how to program stuff, but it is not well put together. And that's me saying that. And you should see some of my code. My code's awful, but that's terrible.
Yeah.
Right.
Was it just like the layout and the formatting of the code? And just like how they tried to solve problems or something? Or did you actually notice
it was how they tried to solve problems, like how they set up their variables, how they try to link things together.
Right there's a hard-coded limit. There's a hard-coded limit of beneficial mutations in the code.
Yeah, I wonder. I'd have to take a look at it, but
it's like you said, assuming the model from the start.
Yeah, if I build a model to test the idea that things can't gain fitness and there's always going to be degeneration, and I code into that a limit in terms of both neutral and beneficial mutations per individual, then that's going to be a problem.
So I have to say, I don't remember where that line was. That could have been setting a default value, but even if it's a default value, why do you have a maximum number of favorable mutations per individual in the first place? That is not a sensible thing to exist.
Yeah.
Right. Maybe the code is test code and they're doing limits testing or something.
Well, the question I had is how did they get from random mutations to basically deleterious effects on a trait that affect fitness? I mean, that is a complicated formula, and
it's probably randomly assigned by a simulation.
Yeah, I mean,
nobody pulled that off.
The random. So they're assuming a distribution of fitness effects, right?
Yeah.
Although they don't actually use a distribution to assign these effects per se. It's sloppier than that.
Yeah.
Wait, how do they assign it? Is it just like completely random? Or what?
I don't remember. It was something like they used a random variable to bin the effects, and then they assigned a value based on some. I think it was just a linear distribution between established values or something like that.
Oh, really? Wait, okay, that's interesting.
Yeah, it was weird. So the one of the biggest things you'll hear this a lot is you'll hear the creationists say, well, everybody knows that you get a lot more deleterious mutations than beneficial ones.
Right.
Right? Mm-hmm. And that's true, because there are a lot of lethal mutations.
Yep.
Like there's a lot of mutations that if you get them, you're screwed. You're not reproducing. Selective effect, 1. Right? Like, so yeah, there's a lot of those, and they don't matter, because if you get it, you die.
They don't accumulate. So, they do not propagate through the population. You don't have to worry about them.
Right. And so if you get rid of those, if you get rid of all of that stuff, it's a lot closer to even. It's still a little bit in favor of deleterious, but we should expect that because most populations are well adapted to their environment.
Right.
So, like, what are we surprised? So the real question is, and this is exactly on point for what they need to be true. The real question is, within the select threshold for effective selection, right? Do you have more beneficial or harmful mutations? Because that we should expect to tell us the real answer. The problem is it's kind of hard to tell because in human populations, or not, in most populations, that value is so small that you can't really measure them. So it's hard to really get into that.
And it's extremely context specific. If you have, like, if you're well adapted for an environment that is extremely consistent, then you're gonna have purifying selection. You're just going to have more of the variation within your population is going to be move you away from that whatever local or global optimal state you're at. If you are adapting to a new environment, then you're going to have a greater fraction that are adaptive in that new environment that you don't already have. It's like, or Sanford is assuming those values are constant and they're not.
Or more to the point, or maybe this is what you said and I didn't understand it right, but to the extent that you have harmful mutations accumulating, these nearly neutral mutations that might accumulate in the population, well, they're harmful for you now, right? But you're also accumulating beneficial ones that are beneficial for you now at about the same rate based on all of the current evidence. And if you go to the distribution of fitness effects of new mutations, the paper they like to cite, great paper, read it a few times, you can see that you have some mutations at 1, bunch of mutations at 1, some at 0.9, some at 1.1. It's about the same. There's a good bit of symmetry there. And so that means that should basically offset itself. And you're not going to get genetic entropy if you're getting the same number of mutations in both directions. Mm-hmm. Well, but more to the point, if it suddenly gets colder or hotter or wetter or drier or who knows what else might happen, right? Suddenly there's new food available. I imagine what you will. You've got a bunch of mutations just sitting there. And suddenly maybe they're selected for it now. Right? And so this is the raw material of it. Mm-hmm. And so again, he's making a point that goes against his point. It's amazing.
Yeah.
Well, okay. And so not even just that, correct me if I'm wrong, but as long as you have, like, you could have no beneficial mutations at all and just have the slightly deleterious mutations. And as long as you're allowing the possibility of back mutation, the population will eventually equilibrate and it still won't go extinct, even without any nearly neutral beneficial mutations. So that it just won't lead to extinction as long as you have a decent enough large population. To maintain an equilibrium.
And you can make that work even without selection. Yeah. You can set up a simulation to do that, and you can ignore selection. And if you start with Sanford's own assumptions, what Sanford claims will happen does not happen. Like it just you set up the dominoes the way Sanford says to set up the dominoes, and then you hit the first one, and Sanford says, "Oh, this is what's going to happen with all your dominoes," and you hit the first one, and like the opposite happens. The thing that he claims will happen will not happen.
Wow.
Yeah.
So
there's oh, go ahead. Sorry.
Oh, sorry. Go ahead.
I was just going to say, there's been an argument that I've been wanting to try because I'm curious what the creationists would respond with. And it's just that the idea, so, obviously Sanford's saying, like, eventually the population's absolute fitness will get to zero, right? Because that's what happens when a species goes extinct. Okay, but the absolute fitness of humans as, like, a whole species is greater than 1 because our population is increasing, not decreasing. So how do they reconcile this idea with, like, okay, your absolute fitness is declining with the fact that our absolute fitness is above 1?
What they'll say, and this is I've had this exact conversation, and what they will say is that, well, you have a bunch of mutations that on their own are not a problem. They're too small to select. They might even be beneficial. But once too many of them accumulate, then they become deleterious. And the problem with that is twofold off the top of my head. One, that's not Sanford's argument, right? And I should say I've had this conversation with not Sanford. I've had this conversation with other people. And what they'll say is, well, you can have beneficial mutations that it's a loss of something. It's a reductive mutation. They'll use, it's devolution, right? It's a loss of something, but it's beneficial in the specific context in which it occurs, so it'll stick around. And also, they will say that you have all these neutral enough mutations, these nearly neutral mutations that are actually deleterious, that when you get enough of them, then you will have a harmful effect on net. And the problems here are twofold. One, that violates the constant fitness effect rule that Sanford has, right? Because Sanford doesn't say anything about, well, it's good now, but it hurts later. He's just, everything is bad, right? So that's one. And two is that in order for that scenario to play out, where you can have short-term benefits and a bunch of neutral mutations accumulating that down the road later on, the combinations become harmful, in order for extinction to result, you need everybody in the population to, at the same time, experience the same loss in absolute fitness.
Mm-hmm.
How's that going to
not
happen?
realistic at all. Yeah,
it
completely ignores things like recombination as well. Yeah.
It can't happen.
And there's one other thing as well, and that is that when Sanford says absolute fitness, he doesn't mean what scientists mean when they say absolute fitness.
Yeah.
Because when scientists say it, they have a mathematical definition, and he doesn't. So.
Right. It's kind of related to that older idea about fitness, or I guess misconstrued idea about fitness of, bigger, stronger, more intelligent, etc.
Yeah, it's similar to, and this makes sense because Sal works with Sanford. So it's the kind of biochemical functional definition that Sal favors, is kind of this redefinition of fitness that to take into account functionality of the genome rather than reproductive effects, right, of a particular allele. That's not what fitness means. You could come up with something else to describe what that is, right? But like that's not what the word fitness means.
But also, sorry.
Yeah, go ahead.
I was just going to say, if you're making up new words and new definitions, you're not allowed to use the framework of population genetics that's established on the other definitions. Like, how are you going to use nearly neutral theory if you're not using the definitions that nearly neutral theory uses?
Yeah. Okay, so I think we've beaten Mendel's accountant pretty badly here. What are we looking at right here?
This is Mendel's accountant. This is where he actually calculates the fitness, and he apparently has some algorithm for calculating fitness based on the alleles. And I'm not sure how deep it goes, but he says basically there are different algorithms for assigning a fitness factor to an initial allele. Pointers to two of them, it shows that initialization time. The thing is that anything they do with this much code has got to be a very naive assignment of fitness to function, right? Or fitness to a set of alleles. And I don't I would like to see the details of how they're doing that. Creo Debunk says he might look into it with Python, and I might help him with it. But we might do a rewrite on this thing and put it into something so that we understand what the algorithms are. Because I got a feeling that he's fudging like hell when it comes to fitness. No, I'm not sure if Sanford and Cell are as naive as Standing for Truth, but the idea of this rusting car, that like for mutations to accumulate, more than one mutation would have to hit a particular gene or affect a particular protein, right? So you'd have to have like if you had an alpha helix that passed through a membrane and you had a mutation of one of them to a polar amino acid, that would have a minor effect on it. It might still work in the membrane. But if you had two or more, but they would have to hit that same gene in order to rust. Now, Standing for Truth thinks the rust is accumulative across the entire genome. What I'm wondering is if they made the same dumb assumptions in Mendel's accountant, whether they're adding them all up, right? I mean, if they're adding them up and they come up with with basically a real number, and they're slowly dropping the fitness down, which would be ridiculous because it's discrete.
Yeah. Well, knowing how like simulation studies generally work, I doubt they're accounting for like an entire organism's genome and all of the fitness effects that because I mean the probability space of that would just fill up so fast that it'd become like computationally impossible.
Yeah, never. We couldn't do it with any supercomputer we have on the planet today in a realistic manner. So I'm wondering. I mean, we can do pretty good simulations, but I doubt that this is one of them. And this thing hasn't been maintained for over 2 years, so I'm really doubting it's. But Standing for Truth calls it the most sophisticated population genetics program ever made, right?
Which is just false.
That's so wrong.
Just
yeah.
I don't even know what to say to that. Like,
what do you say to him?
I mean, like the degree to which you had like the confidence to knowledge ratio that is required to say that sentence with a straight face is just astonishing.
And knowing this, know that he has never downloaded this program and ran it.
Or dealt with any population genetics program.
Yeah.
All right. So I think we've with one. There's one more thing I want to do with Mendel's accountant before we cover the rest of this conversation. And Speed, could you pull up, Professor Evo, could you pull up that peaceful science thread one more time? Because I want to people were very helpful in that thread and put in the parameters for real experiments that have been done into Mendel's accountant to try to see if it will replicate the real results of those experiments. So if you scroll down to post number 36, if you just scroll on the right, it has the numbers 24 out of 84. Just keep scrolling. It'll get to number 36. You're going really fast. Hang on, what's going on here?
Where aren't they updating?
Oh, it's not updating. Oh, that's not there. Stop right there where there's red and blue.
Okay.
Where's that red and blue figure right there? Okay, because that you're in the frame there. There you go. Okay, so that is Mendel's accountant running the Lenski long-term evolution experiment over, in that case, 17,500 generations. And the useful thing to look at there is the red line is relative fitness, right? And you can see that there is a slow but significant decline in fitness over the course of this simulation. And this is running this with the realistic experimental parameters in terms of things like population size. So scroll up a little bit, because we also have in this same thread, it's a figure that has two curves, one in green, one in black. Keep going. It's above that. It's right there. It's that one. It's above. It's the one there, right there. The one above it.
Oh, okay. There's a little bit of
that one.
Little bit of a delay.
That's the real one. Those are the real data showing a significant increase in fitness over the course of that experiment. So you can't even, by putting in the actual parameters, you cannot make Mendel's accountant replicate the actual experimental results.
I thought the relative fitness was the one that was below that a little bit. So,
no, that's the LTE.
Oh, okay, right. Sorry.
Yeah, that's like the one where it's the Yeah, the big increase initially, and then it kind of slows down.
Like, what you fail to understand, Dan, is that this Mendel's accountant is far more sophisticated than E. coli.
It's far more sophisticated than the actual experiment. Yeah.
Yeah.
Mm-hmm. Yeah. So that's the last thing I wanted to show everybody when it comes to Mendel's accountant, because like we have experiments that have done this, and you can't make Mendel's accountant match the experiments. And there's one other actually funny thing that someone in this thread on Peaceful Science mentions, is that if you put parameters for humans in there, with 8 humans getting off the ark, it always results in extinction.
Yeah. So,
good. So hang on a second. I do want to point out that everything we're showing here, including the source code and everything, is linked in the description, along with all the papers that SFT pulled up, plus the original video that where you were chatting with SFT. So,
okay.
All right. So
you know how I love my descriptions.
So I have
there's a lot more in that conversation. So Andrew, where are we going next?
So this kind of gets into some other responses and some actual questions I had for you, Dr. Dan.
Awesome.
Which was basically this. I've kind of talked with or emailed some other professors on this, and one of the things they said was that you either have, like, along the lines of, you either have a neutral mutation or it affects fitness and selection acts on it, like in some way, at least in the normal size population. So I guess I'm kind of confused how they can even claim that they're effectively neutral but also slightly deleterious. Like, how does that even make sense?
So that's another huge. Dr. Dan can answer that if you want.
No, go. You go.
Okay.
You're really good at explaining this.
So that's like one of my favorite misconceptions that a lot of creationists have. They think that, like, if you get to the selection coefficient cutoff, right, there's absolutely no selection on a specific allele, which is just dumb, because the point of nearly neutral is that it has a fitness effect. The idea of nearly neutral is that when you're below a certain selection threshold, the effect of drift will be stronger than the effect of selection. Right? So,
and it becomes probabilistic at that point, rather than just selected for or selected against.
So basically this only works for smaller populations. In like normal size, for larger populations, like selection is a bigger factor, basically.
So the technical thing is that in a larger population, you can have selection operating on a smaller, what's called the selection differential. The difference between the individuals with a particular allele and the average for the population, that difference in fitness is called the selection differential. And the larger the population, the smaller you can have that interval be and still have selection, right?
Mm-hmm.
So
okay, that makes sense.
So whether you have selection or drift kind of driving the changes in frequency for a specific allele is going to depend, and I'm oversimplifying this, but it's going to depend primarily on the population size and that selection differential, right? So you can have a small population, but if you have a huge selection differential, then you can still select for it. If you have a really big population and a really tiny selection differential, you can still select for it. It depends where you fall in terms of those two things.
And may I ask what the selection differential depends on? Does it depend on any other factors?
It's the average, so it's relative fitness, basically. So it's the average reproductive output of the individuals with a particular allele versus the average for the population as a whole.
Okay.
Yeah. You can sort of think of it as like, so selection and selection coefficients are already normalized metrics, right? They're already normalized to whatever your population is. But the effect of randomness is dependent on the total size of the population. The way I sort of think about it is like, let's say you have this curve on a graph that's it's a perfect selection coefficient, right? It's a graph of all of the different possible selection coefficients. And the population size that you have is going to try to approach that curve. And so if you have a really big population size, it'll match that curve really well. But if you have a really small population size, it'll staircase it to approximate it, right? And so, when you have a smaller population and some random act happens, like let's say you have 10 people, and I don't know, dude gets hit by a car or something, right? That's going to have a much larger effect on the total amount of alleles available in that population. Whereas if you have, 1 million individuals, the effect of those smaller acts aren't going to have as big of an effect on the allele frequencies.
Right. Makes sense. So this kind of gets into one of the other responses SFT put to the Bristol question, which was basically that genetic entropy works on the population level. So basically, this individual example isn't relevant to genetic entropy. Something along those lines.
What does that even mean?
I don't know what he means.
If it's a new allele, it's in the population.
I don't even know what that argument is. It's genetic entropy is about it's a population genetics argument. So when you introduce new alleles into a population, that's relevant to the population genetics of that population.
Right.
So like individuals have alleles, and then how those alleles change in frequency is going to be the result of parameters at the population level.
It's kind of like zooming into the trend they're claiming, like this line. Basically, you zoom into a specific piece of it, and they start throwing up their hands in confusion.
Yes, I mean you'll notice that throughout this entire conversation, none of anything that SFT and Godzilla Freak said addressed the fundamental shortcomings and contradictions in the genetic entropy argument. My suspicion is that they do not understand population genetics sufficiently to realize that those contradictions are there. But like none of the stuff they said was like referencing any concrete population genetics concepts. It was just a bunch of different ways of saying that harmful mutations cannot be selected out and therefore they accumulate over time. And like, okay, you need to like show the math on how that works.
Right. Like they keep made the claim basically that the information content didn't change, but the total functionality is still decreasing for some reason.
So now we have three things, right? We have fitness, we have information content, we have functionality. They're all different, but nobody can tell me how they're all different, right? Like, what are we doing here? So, the other kind of problem with a conversation like this is, and I'm going to specifically pick on Godzilla Freak for just a minute, is that they don't know when, like, what, like, specific terms mean, and they conflate things that are the opposite of each other. So, like, for example, Godzilla Freak at one point said that even if you could select out these harmful mutations, that would be a problem, right? He's saying that like genetic entropy would still happen. You'd still go extinct because how do you select things out? You kill individuals. Because every individual is getting all the bad mutations, you select them out, the individuals die, and that's genetic entropy. Like,
Wait, so is he saying that like every single mutation gets fixed into the lineage, unless that lineage dies?
Yes.
I forget how he said it. I wish I could find the transcript on that piece because he said something that was really ridiculous.
So Walker, what you're trying to do is like describe the population genetics of what he like translate into like actual population genetics speak. I don't think you can do that.
Okay.
Like I was trying to explain to them, like, if you have even a slightly deleterious mutation in this large population, for example, that lineage like is going to have lower fitness, meaning it's going to produce less viable offspring compared to others. So it's not going to propagate as much, and it's not going to become fixed. Like,
and that's literally the definition of selection. What you just described is literally what selection is, right?
Well,
so,
and also, all right. So let's just say that let's pretend like in an alternate universe, like selection, there is no selection coefficient of nearly neutral mutations. Like there's no selection acting on them at all. Do you know what happens to like 99% of neutral mutations? They drift out of the population because every single time you reproduce, there's only a 1 in 2 chance that it gets passed on. Just because, you only give, unless you're assuming that you give your children all 46 of your chromosomes, which would be interesting. But no, you only give half of your alleles to your offspring, right? So if you're just looking in like a straight line, obviously organisms will have more than one kid. But if you're just looking in a straight line, there's only a 50-50 shot it gets passed on in one generation. There's only a 50-50 shot it gets passed on the next generation. And the probability that it drifts out of that lineage grows exponentially with each generation.
Even with a bunch of kids, if you use like a high estimate, even if you assume 10 kids per person in that idea, it still mostly drifts out.
Yeah.
Right. I think the misconception, like at least if I'm understanding what he was saying correctly, or what he was trying to say, was I kind of think of it like he was basically saying, okay, you have a say these imaginary creatures, for instance, and they're all different colors to represent the different selectable variations, for instance. And if you have even a slightly harmful variation, then that individual immediately dies. Whereas, for instance, sometimes it can be passed on, but it doesn't get passed on as much, and so it still doesn't. So either way, it doesn't get passed on. So that's kind of the major thing here. Like, whether it dies or not is kind of not entirely irrelevant, but am I talking about this correctly?
If it dies, that makes selection work real well.
Yeah,
Right.
Selection's working really fast. If your selection coefficient 0.
Was it right here, Dr. Dan, where Godzilla?
Well,
I can always tell when you're talking, Andrew, because I see the word sorry. Because you're always apologizing.
Most of the so that is a different place where they say that most but we can have natural selection, the best beneficiation. Most of them are reductive. Yeah, that's not true. Like, they don't have the data to back that up.
Well, like they keep bringing up this example of a 1 in a million, for instance. Is that even true?
That's also BS. They don't have the data to back that up.
Okay.
Natural selection. Yeah, there we go. And we'd still be extinct, right? That's right there, right? So they're saying, yeah, it's either way. So now this is a bit of having your cake and eating it too, right? Because saying, well, genetic entropy says no selection, therefore extinction. But even if you had selection, therefore extinction, it's like, well,
which is
heads I win, tails you lose, right? Like,
well, he's claiming that the entire population would be selected against. The entire population is what he's basically
which
saying,
is a contradiction in terms because of the concept of relative fitness.
Right.
You can't have literally can't have the whole population being selected against. There's nothing to select against.
I mean, I do kind of disagree with that. I think everybody has deleterious alleles that are selected against, but as an individual, you're not necessarily being selected against.
Right. That's as an individual, not every individual within a population can simultaneously experience negative selection, because whether or not you are experiencing positive or negative selection is based on your reproductive output compared to everybody else. It's like, oh, we want all the kids in schools to be above average. Well, someone's going to be below average, right?
For human beings, there's one other thing that they frequently gloss over, and that's that when the sperm is finding an egg, basically at that zygote level, there's a whole lot of natural selection going on when it comes to severely deleterious mutations, and a lot of things don't even make it to the pregnancy point. Yeah. And if they do, you don't even know that you're pregnant, and so anything that becomes that is truly deleterious, like when the rust on the car, as SAT would put it, gets to the point where the fender's about to fall off, you don't even know that you were pregnant. The fender falls off at the zygote stage. So and that's
can you put meltdown in your little search box there? Because that was in my notes, kind of after where we are. There we go. It's right there. Mutational meltdown. Right. This is another one that SFT uses. He goes into this thing of mutational meltdown, and it's related to this, what we're talking about here. And the problem is that this is another phrase that has a very specific definition. Mutational meltdown is what we were talking about before. It's what happens in an extinction vortex, where you have deleterious mutations that are recessive. And due to inbreeding, those recessive alleles accumulate. Due to small population size and inbreeding, you genetic drift, You get these recessive alleles accumulating, and that leads to a loss of fitness and ultimately extinction. So that, in terms of what's going on in the genomes of the population, is the opposite of what Sanford claims is happening.
Yeah. Now, I had another document. This thing about, oh God, there's so much to cover. Increased rates of autism. Okay. I looked into that a little bit, and there's an increased rate of diagnosis of autism, but there's no indication of an increase in autism. Decrease in IQ. There's a bunch of studies that indicate that that's not happening at all. There's actually an increase in the number of.
There's a net increase in IQ.
Yeah.
And cancer peaked in the 1980s and has been dropping ever since. And I'm not talking about the cure or the death from cancer. I'm talking about getting cancer. And I pointed out that it seems odd that we put catalytic converters on cars and we quit smoking a lot, and the rates of cancer went down.
What a
the only one that's still up, I think, is prostate cancer.
That's just because people are older.
Yeah, and it's not actually. Well, it's not actually going up. It's flatlined. Basically, it's flatlined.
No offense, Speed, but you've probably got some cancerous prostate cells. You're old enough.
Oh, I do not. I'm not that old.
It's no big. You're over 70, right?
Actually, I'm going to a doctor next week to check this out.
Oh, that's good.
Yeah, it's just that's when you hit 70.
Everybody should get your prostate checked
probably
once over 40.
Yeah, it's the second thing to go, or no, the first thing to go.
You're not. If you get your first cancerous prostate cell at 70, that is not what's going to kill you.
Yeah.
Yeah.
So on the subject of humans, this seems to be this example of humans seems to be their main example of genetic entropy, even though I tried to point out to them.
Yeah, go ahead. Go ahead. But this line is laughable, and they say this all the time. But keep going. And
I know. Go ahead.
So, humans are kind of a terrible example because we're not the same. Maybe there's selection going on, but we're not the same level of selection as natural populations. So it's kind of not analogous.
I would put a qualifier on that, right? A lot of the things that we would typically view as deleterious or something are things that don't actually matter to our reproductive success. So your tonsils or prostate your wisdom teeth or something like that. Yes, people have messed up wisdom teeth, but since we have the ability to get them taken out pretty easily and they almost never result in deaths before someone's able to reproduce, that would be an example of an effectively neutral mutation.
Yeah,
right. It's probably just going to drift around in the population.
And even if humans were a little bit weird and we had something going on, and Kondrashov's whole book is about the worry that basically we are screwing up natural selection and we're accumulating mutations. Even if that's true, it says nothing about evolution at all.
Right.
Nope.
Yeah.
No.
Yeah.
So can we just comment on this line real fast here? Because this is something that creationists say all the time, is that we're seeing the rise of thousands of new genetic disorders.
No.
That is not true.
No,
what we're doing is we are finding more as we're doing more comparative genomics. But we're not seeing an increase in all these novel genetic disorders that have never existed before. What we're doing is we're finding the genetic basis for things that have existed. And now by doing comparative genetics, we're able to figure out if there is a genetic basis for it and what those alleles are.
I'm glad you said that.
There's an infinitely large increase in genetic-related disease conditions being spotted between now and 1940, for instance.
Amazing, right? It's like they didn't even exist prior to the completion of the human genome. Who would have thought?
Yeah. One other thing that SFT brought up is he said that all mammals are basically, what was it?
That's my next thing, with the same mutation rates, right?
With the same mutation rates.
Okay.
Okay, so Kumar, he cites Kumar in 2002. He wasn't talking about that. In 2005, Kumar pretty much says the opposite in that paper. And I had a Word doc up that was from a page that I also linked, and I'll try to put that up.
The problem with this whole thing about similar mutation rates in the example that everyone loves to use is humans and mice. The problem is a conflation of mutation rates and substitution rates. And this is one of the things that just nails on a chalkboard for me. A mutation rate is measured as mutations per site per replication. How many mistakes do you have every time you copy the DNA, right? What's the frequency of errors when you copy the DNA? The substitution rate is substitutions fixed, right? Per site, per year, right? So it's normalized per year, and it's only talking about changes that are fixed in the population. So the important thing here is that the substitution rate removes generation time as a factor. Now, if you read the paper that they talk about here, they say mutation rate, but then they say mutations per site per year. So what they're describing is substitution rate. And here's the important thing. The mutation rate for mammals, the per base per replication mutation rate for mammals is pretty similar. All mammals, it's within I think it's within 1, maybe 2 orders of magnitude in terms of genetic scale, it's close enough, right? It's close. The substitution rates are even closer. And that's where it gets weird because mice have a generation time of what? 6 weeks, 2 months, something like that. It's short.
Yeah.
Right. So if you have mice with a generation time of 2 months and you have humans with a generation time of 20 years, well, those mice should be going through more mutations in that same amount of time. But if they're only fixing the same number of substitutions approximately, or they're fixing substitutions at approximately the same rate, what is responsible for that discrepancy? Selection is responsible for that discrepancy. Standing.
I mean,
that proves you're wrong.
It proves selection, and it shows you that genetic entropy does not work at all for mice.
Bingo.
And there's something else. I did a back of the envelope calculation on old world monkeys, which I think are mutating 30% faster than us, and that shows that they went extinct in 200 AD. Basically, there are no old world monkeys because, according to the young Earth creation timeline. So, anyway, they went extinct 1,800 years ago. According to Sanford, he says that we're going to be extinct in 300 years. So I just back calculated. So, anyway.
Wait, how did he derive the number of 300 years?
How did Sanford come up with 300 years? I think he figured. Doesn't he say that in his book somewhere? Or what?
He's going by generations from. I think he goes from the arc. I forget if he goes from arc or creation, but it's one of them. And then he goes mutations per generation. Functional fraction of the human genome is approximately 100. Yeah, that's another thing he says. By the way, that's another thing he says is that the functional fraction of the human genome is approximately 100%.
With a straight face.
He says that. That is, and people have the funniest thing, and I've actually gotten into arguments with evolution people about this because I will be describing Sanford's argument, and they'll be like, "No,
it's not that bad.
That's not." And I'll be like, "Did you read the book?" And they'll be like, "No." And I'll be like, "Here's the part of the book where he says the human genome is approximately 100% functional, and approximately every mutation is going to be harmful." He says it right there. And so what he does is he calculates the mutations per generation, the functional fraction, generation time, genome size, and he uses the drop off in lifespans in Genesis as evidence for this. And he says you can plot this out to where we're going to go extinct.
Yeah. Old world monkeys went extinct to 200 AD.
The appropriate thing to end that sentence with, by the way, Dan, is you can't make this stuff up.
Right. Yeah, it's bad.
Yeah, I think Carter's very inconsistent about what species are and aren't experiencing genetic entropy. I'm pretty sure he's I think I heard him say humans are definitely experiencing it. Mice are definitely experiencing it. Bacteria aren't experiencing it, and then viruses are.
H1N1 definitely is, but H1N1 definitely is, but HIV is not.
Yeah, it's so weird. It's so inconsistent.
And bacteria are not. And the reason why bacteria are not is because they have such fast generation times and large populations that they can weed out the bad stuff. And it makes me think, I think there's a word for that.
I have to say, I absolutely agree with that assessment. Bacteria have sufficiently large population sizes, and they reproduce enough that they can avoid this.
They can do selection. Yeah, it's weird.
And
right?
all other normal populations.
And there have been some papers on occasionally their mutation rate is increases, and I think the Lenski cultures had that happen a couple times.
There's one of the lines became a mutator phenotype. Yeah, one of the 12. Yep.
Yeah, which makes them survive rather than kill them.
That line, the mutator line of the Lenski experiment, has the second highest fitness after the citrate line.
Yeah.
So that kind of raises the question of, if on net mutations are bad, and the more mutations you get, the worse you will be, and this is what Sanford says very plainly, then why is it that the line that adapted to have faster mutation rates is the second most fit out of the 12 in that experiment? Because bacteria don't experience genetic entropy for reasons.
Yep.
So I'd like to take a vote real quick before we move on to another topic, if we do. So everyone in the room, I'd like to hear your thoughts on this. Do you think, or just yes or no, do you think SFT and Godzilla Freak adequately and sufficiently addressed the bristle question the other night?
No, the part where Godzilla Freak was saying, oh, so the bristles disappeared from one spot and popped up in another spot, so there's no new functional information. Well, now, if you were to be able to calculate functional information, and you can only do it right now with enzymes and make any sense out of it, that would be a different amount of functional information, because we're talking about function, right? So if the bristles popping up in another spot had some effect on that population or some effect on survival, then there would be a difference in functional information. But the way he put it, just because the bristles disappear from one spot, pop up in another spot, no new information according to them. But they have a really bad relationship with information, with that word. It's very loose. And when they think about it, Maddox is currently going on about fits, or functional information bits. And we did a little video on that, and I took it down because I didn't care for how it went. But anyway, you can calculate the functional or fit, what's called functional information bits or fits on an enzyme by taking the fraction of conformations and dividing it by the total number of possible amino acid combinations for an enzyme, right? And you'll come up with some number. Well, you take the log 2 of that and make it negative, and you'll come up with the number of bits of information. And they've done it in a couple of papers, and I don't know what use it is, but it tells you like you'll have 14 bits or you'll have 8 bits. But now if for instance 25% of the protein conformations do the same job, then it's 1/4, and you have very few bits of information. And there are ways to do it, but they never do any of that work, right?
Yeah, that sounds like that Douglas Axe paper from like 2005, where he takes one specific protein and he's like, look how unlikely this functional conformation is. Oh, not realizing that there's hundreds of like similar sequences that will generate that same function.
But he did one in 1998 for Barnase, I think, is a protein, or Barnase is a protein, and he came up with, surprisingly, 1 out of 4 mutations that they threw at it were functional. So 25%. So you've got 2 bits of information there. That's it.
Yeah, that sounds about right. Yeah.
So
go ahead.
So it seems like, though, then this is, rather than quantifying information, it seems like it's quantifying constraint.
Well, what it does, it takes like for Shannon information, you just take the log 2 of the number of bits that you have to run through. Like for every bit, you make a decision. I'm going to go left or right, right? So if you have 2 bits of information, that means you can divide a population of proteins into 4 different chunks. So that's 2 bits. If you have 8 bits, then you divide it again. So you make another decision in order to get there. And, it's a number that they're using, and I have a paper on it, and I should put that video back up again. I should edit it. But anyway, it gives you a little bit of information, but not a lot. It just tells you how many proteins in the protein space will perform the same function. So that's it. Usually it's around 9 or 10 bits or whatever of information, which is a hell of a lot. But there are reasons why proteins evolve the way they do. So anyway, I got interested in it.
So Dr. Dan,
compare that number against the amount of actual information in the protein, in a protein of the same length, right?
Yeah.
And you just get a ridiculously tiny fraction.
Oh yeah.
Andrew, before we continue on, can I just we're almost up to the end. We had gotten to like 32, 33 minutes in the transcript, and that was or like 35 minutes, and that was pretty close to the end of where you switched and Brenda came in. There was one more little bit that I noted though that I thought was funny, because this is the 3rd time at least I've heard SFT say this. He said it to me. He said it in Walker's debate on Neanderthals. Then he said it again here. And it's the inbreeding leading to more mutations thing.
Oh god.
And that was it was somewhere after 35 minutes. Maybe they're inbreeding. It's faster mutation accumulation and accelerated genetic generation. Since we're all multiply, so he's talking about inbreeding here. And I think if you go down a little bit, he goes on and he did the thing with inbreeding where he still seems to think that what happens with inbreeding is you somehow get a faster mutation rate, and it's and that's not what happens. Yeah. That's not what inbreeding does.
Doesn't it have to do more so with like recessive alleles and things like that?
It's that you're with inbreeding, you are increasing homozygosity. Right. And that results in the accumulation of recessive phenotypes, right? Because you're increasing the likelihood that 2 individuals share an allele, right? Yeah. They're homozygous due to having recent common ancestry, right? So you've got great-grandpa up here, has lots of kids. They all inherit great-grandpa's one recessive allele, but then you've got like second cousins having kids down here. So the kids inherit the same allele from both parents, and you end up homozygous recessive. And so you get more recessive harmful phenotypes in situations with inbreeding, but you're not getting more mutations. And he's talking about faster genetic degeneration here. And you're not accelerating the mutation rate. It has to do with the combinations. And like, I don't know how to make him not get this wrong every time.
Yeah. You're actually getting less mutations because there's less individuals in the population that are contributing to the amount of variation.
You're decreasing the effective population size.
Yeah, and there was a paper a while back. It was kind of surprising, but they pointed out that for cousins, apparently, there can also be a very positive effect of the same thing. I mean, you can end up with 2 beneficial mutations that become dominant much faster. But
I got to point out that SFT also thinks that if you have a woman living closer to somebody of a different race, that the mutation rate in her ovaries will go up.
Oh my God! He said that out loud.
Wait, where?
No, he didn't say that out loud. It's just the necessary implication of things he did say out loud, because he thinks that the mitochondrial mutation rate increases when the population diversity is higher.
Right.
So,
he thinks
explain to me how else that can work.
He thinks Neanderthals basically ran into the Ice Age and they mutated faster because they were more stressed or something.
This was the thing. Well, this was the thing that Walker got him on in the debate, where he's like, you had this inbreeding problem that led to mutations that like broke DNA repair or something, and that leads to a higher mutation rate. And Walker asked for a specific example, and the example he gave was the opposite.
My favorite part of the debate was in the first 15 minutes or so, where he said that, right? He was like, we would see the effects of inbreeding in autosomal DNA, but in the mitochondrial DNA, it's free to hypermutate or whatever. And I pulled up a paper. I showed him. I still have the paper. It's skeletal anomalies in a site in Spain. I forgot the name of the site. Where they were like, we sequenced all of the genomes and we found inbreeding through sequencing their mitochondrial DNA. We found close kin relationships because all of their mitochondrial genomes were super closely related. Right? You can still see you don't see hypermutation anywhere, and you see inbreeding everywhere, basically.
Yeah.
All right, so we've been going for like an hour and 15. What's our target here?
Yeah, so I want to ask one more thing, Dr. Dan, before we wrap up. Kind of, since we I can't even say. Since we pretty much debunked genetic entropy and their responses to the bristle question, do you think I should still harper on them with this, on the bristle question, or should I leave it alone for a while? What do you think is the next step to go with this?
Well, go ahead.
What?
Jackson Rowe last night mentioned 16 de novo mutations that increased fitness in E. coli in the gut last night. And that would be an interesting thing to harp on them about for the next 6 months.
I mean, my thing with genetic entropy is just like, show me how the population genetics is given what Sanford says, right? Fixed fitness effects, no context specificity, constant accumulate, show me how you can start with all those conditions and get the outcome that Sanford claims. And just show me how those things are not self-contradictory. And I don't think there needs to be more of an argument with genetic entropy until they can show not even specific cases, but just show the basic math of if you plot out these situations on a graph, is it supposed to go, what's the dependent variable, and is it supposed to go up or go down? And why? Right?
I want to know what's meant by accumulate.
Right. Show me how it's supposed to work, because Sanford wrote a book on it, and at no point is there enough math in this book to write out the equations and plot them in such a way that explains how it's supposed to actually work. CRISPR's favorite thing to harp on is that population genetics as a field is math, right? And there's a joke that population geneticists are just mathematicians who like biology, right? It is math.
Yeah.
So make the argument based on math. Don't tell me about rust on a car. Don't tell me about how, our genome is deteriorating. We're getting 100 mutations per generation. Show me how what you claim must happen actually is supposed to happen. Show me the math in terms of how that's supposed to work. And the obligation is on the person making the argument to do those things. And if they can't do those things, then that's the end of the discussion. You can't.
They don't even have a model at this point.
You can't support the affirmative claim that you are making. Okay, then I don't have to entertain it. I don't have to take it seriously. You can't back it up. Have a good night, right? That's my thing with genetic entropy and all the specifics of it, of we have 100 mutations per generation and how much of the genome is beneficial and H1N1. We didn't even talk about H1N1 tonight, but that's a whole other thing. And all that stuff is a sideshow, because the fundamental model that they are supposed to have doesn't exist.
Right.
And that's the end.
It doesn't fit reality.
Nope. Cool.
Yeah.
Skewered. Oh, but what will they say about this stream?
Yeah.
No.
Probably that we're forced into early retirement or something.
Yeah. Anyway, okay.
Should we wrap up?
Well, we can wrap it up. We can shoot it in the head. We like to shoot our streams in the head around here.
Wrap it up before you shoot it in the head. It's
okay.
more
Yeah, I
sanitary
just want to
for
say
fun.
again, thanks, Dr. Dan. Thanks, CRISPR. Thanks, Walker, for coming on, adding to the stream, providing insights, all that cool stuff.
And all of you guys stick around for just a minute after we go backstage. Okay, don't just run off. Anyone else have anything they have to say about anything upcoming?
I have a video coming out tomorrow at 6:30. Actually.
Holy
really?
shit.
Are you
I
teasing?
do.
You're not teasing us, are you?
I'm not. It's already set for a premiere.
Oh
Yeah.
man, what time? What time?
6:30.
Cool.
What time zone?
Oh, 6:30 CST.
Okay,
So
cool. That's awesome.
You should have. Did you give me a link?
I can.
Okay, that would be
It's on
good.
my channel.
Because I'll put it in the description of this video, and we should put it in chat at some point.
That's how you get on
Just throw it in the public
website is you bring in links.
Yeah. So today was my 1 year anniversary. My first video was loaded on this channel a year ago. So
Congrats.
The funniest thing actually is my 1 year anniversary was yesterday. So we
I know,
started our channels at almost the exact same time.
yeah,
Nice.
yeah.
Nice.
I've been getting
Very
one
nice.
sub. I've been getting 1.005 subs a day. Something like that.
When you say it
You
like
lost
that,
me
it makes
something,
me
Dr. Dan?
much more sad.
Yeah, I'm sad. I'm very sad. Okay, guys.
Dan, how old is your channel?
My channel,
Younger.
is it younger? It was a
Yeah.
pandemic thing, so I don't know. I don't remember, but it was sometime
Yeah, I think
pandemic that I started it.
June
Yeah.
or July, I think.
I would like
It
to
was
say
summer?
I started my channel explicitly because I felt like there weren't people talking about the genetic arguments that creationists make. And then as soon as I made my channel, Dr. Dan makes his channel and just does it better.
I know.
So,
Well, it's a we're but I will say that your genetic entropy video, Walker, is really awesome and got a crap ton of views and is really, really
it's
good.
true. I
So
think I
it's-
might remake it though, because honestly,
Yeah.
I argued against a completely different topic. I feel like I was trying to push the point that like, yes, beneficial mutations do happen. When I don't even have to show that, I just need to show what genetic entropy doesn't make sense, like in its own merit.
15 ways to show genetic entropy is wrong. So I do have a slight update. I guested on Paulogia's channel. I did a thing for him. It came out on Monday. So you haven't seen that, check that out. Talk about Haldane's dilemma. And that was cool.
Yeah.
That's it. Oh, and I'm doing something next week with Carter just put out a video that's Rob Carter, not James Carter. There's two Carters. It's confusing. Rob Carter just did a video, just kind of a catch-all creationist genetics thing, and a little bit of human evolution in there. So I was watching it. Walker was watching it. Guts Gibbon was watching it. We're all in the chat at the same time. Kind of having, throwing rotten tomatoes during this video. And so we're going to talk about that next week, and that'll be fun.
Now, who's
It was
we?
bad. It was dumb.
Cool.
Who's we and on whose channel?
Oh, it'll be on mine. I know Erica's in. Walker, if you want in.
Depends. I'm incredibly busy. I'm moving back this week for the summer. So
Well, if you're around.
Oh, let me
I'll look
get
forward to that.
Okay, I'm putting the URL for that Paulogia thing in the chat
Oh, thank you.
with you. And okay, we can kill it. Oh, by the way, because my production values are shit, and Dr. Dan and Erica, we actually hate you guys because you make us want to quit our channels because you're so damn good.
Oh,
Mine are terrible. Mine are awful.
No, they're good.
There's
They're really
nothing.
good.
There's no animated stuff. I do nothing for mine. It's literally
The
exactly how I do
information-
lectures for class.
The
Anyway,
information hooks a
So
person.
The trick
the
to
value-
it is you just
Go
have to have still pictures with transitions, and people think they're animated.
I know.
that's what I do. You don't have to call me out like that, Crispy.
Anyway, I think the value of this channel, if you watch the weather report and stuff, is that when I'm on the ball, like this video, for instance, has a great description with a lot of links to papers. And if you ever refer back to it, you're going to be able to pull up all the papers and all of the associated documents. And so it becomes a good resource for that. So I hope I serve some function. So anyway, okay, I'm going to
Thank
kill
you,
it.
Speed.
Okay, goodbye, cruel world.
Thanks for